Hey everyone, welcome to the video lecture for Unit 3, Module 6.1, where we'll cover a basic overview of the lymphatic system and some structures of the body's defenses. As always, here are your learning objectives, so be sure to utilize these when you are studying for both the lecture quiz and the unit exam. All right, let's go ahead and get started. So, many organs and systems work together to keep us alive and healthy. The lymphatic system plays a central role in the ongoing struggle to maintain health. In this unit, we discuss the components of the lymphatic system and the way those components interact. So the world is not always kind to the human body. Accidental bumps, cuts, scrapes, chemical and thermal burns, extreme cold, and ultraviolet radiation are just a few of the hazards in our physical environment. Making matters worse, the world around us contains an assortment of viruses, bacteria, fungi, and parasites that are capable of not only surviving, but thriving inside of our bodies, and potentially causing us great harm. These disease-causing organisms, called pathogens, are responsible for many diseases in humans. Each pathogen has a different lifestyle and attacks the body in a specific way. For example, viruses spend most of their time hidden within the cells, which they often eventually destroy. Some of the largest parasites actually burrow through internal organs. Many bacteria multiply in interstitial fluids where they can release foreign proteins, enzymes, or toxins that can damage cells, tissues, or even organ systems. And as if that were not enough, we're constantly at risk from renegade body cells that have the potential to produce lethal cancers. The lymphatic system includes the cells, tissues, and organs responsible for defending the body. This system acts both against environmental hazards, such as various pathogens, and against internal threats, such as cancer cells. These cells are vital to the body's ability to resist or overcome infection and disease. Lymphocytes respond to invading pathogens, such as bacteria or viruses, abnormal body cells, such as virus-infected cells or cancer cells, and foreign proteins, such as the toxins released by some bacteria. They act to eliminate these threats or render them harmless through a combination of physical and chemical attacks. The ability to resist infection and disease is called immunity. We have two forms of defense systems that work independently or together to provide immunity. These forms are innate, or nonspecific defenses, and adaptive, or specific defenses. Surface barriers prevent or slow the entry of infectious organisms, or attack them if they do enter. These defenses are called innate because you're born with them, and nonspecific because they do not distinguish one potential threat from another. In contrast, lymphocytes respond specifically. If a bacterial pathogen invades peripheral tissues, lymphocytes organize a defense against that particular type of bacterium. For this reason, we say that lymphocytes provide an adaptive defense. Defense against specific antigens is known as the immune response. All the cells and tissues involved in producing immunity are considered part of the immune system. The immune system includes parts of the integumentary, skeletal, lymphatic, cardiovascular, respiratory, and digestive systems. And we'll begin this module by examining the organization of the lymphatic system. We'll then consider the body's two forms of defenses, and finally, we'll see how the lymphatic system interacts with cells and tissues of other systems to defend the body against infection and disease. The lymphatic system consists of one, lymph, which is a fluid that resembles plasma but contains a much lower concentration of suspended proteins. Two, a network of lymphatic vessels, often called lymphatics, which begin in peripheral tissues and connect to veins. Three, an array of lymphoid tissues and lymphoid organs scattered throughout the body. And four, lymphocytes and smaller numbers of phagocytes and other cells. Primary lymphoid tissues and organs are sites where lymphocytes are formed and mature. They include red bone marrow and the thymus gland. Recall that red bone marrow is also where other defense cells, like the monocytes and the macrophages, are also formed. Secondary lymphoid tissues and organs where lymphocytes are activated and cloned, or production of identical cellular copies. These structures include the appendix, the spleen, lymph nodes, tonsils, and mucosa-associated lymphoid tissues, or MALT, in the digestive, respiratory, urinary, and reproductive tracts. The primary function of the lymphatic system is to produce, maintain, and distribute lymphocytes that provide defense against infections and other environmental hazards. To provide an effective defense, lymphocytes must detect problems and they must be able to reach the site of injury or infection. Lymphocytes, macrophages, and microphages circulate within the blood, and they're able to enter or leave the capillaries that supply most of the tissues of the body. Capillaries normally deliver more fluid to peripheral tissues than they carry away. The excess fluid returns to the bloodstream to the lymphatic vessels. This continuous circulation of extracellular fluid helps transport lymphocytes and other defense cells from one organ to another. In the process, it maintains normal blood volume. It also eliminates variations in the composition of interstitial fluid by distributing hormones, nutrients, and waste from their tissues of origin to the general circulation. Lymphatic vessels carry lymph from peripheral tissues to the venous system. The smallest lymphatic vessels are called lymphatic capillaries. The lymphatic network begins with lymphatic capillaries or terminal lymphatics, which branch through the peripheral tissues. Lymphatic capillaries differ from blood capillaries in several ways. They, 1, originate as pockets rather than forming continuous tubes. 2, they have larger diameters. 3, they have thinner walls. And 4, typically have a flattened or irregular outline and sectional view. Lymphatic capillaries are lined by endothelial cells, but the basement membrane is incomplete or absent. The endothelial cells of a lymphatic capillary are not bound tightly together, but they do overlap. The region of overlap acts as a one-way valve, and it permits fluids and solutes, including those as large as proteins, to enter along with viruses, bacteria, and cell debris, but it prevents them from returning to the intercellular space. Lymphatic capillaries are present in almost every tissue and organ in the body. Prominent lymphatic capillaries in the small intestine, called lacteals, are important in the transport of lipids absorbed by the digestive tract. Lymphatic capillaries are absent in areas that lack a blood supply, such as the cornea of the eye. The red bone marrow and the central nervous system also lack lymphatic vessels. From the lymphatic capillaries, lymph flows into larger lymphatic vessels that lead towards the body's trunk. The walls of these vessels contain layers comparable to those of veins. Like veins, the larger lymphatic vessels also contain valves. The valves are quite close together, and they produce notable bulges. As a result, large lymphatic vessels have a beaded type appearance. The valves prevent the backflow of lymph within the lymphatic vessels, especially in the limbs. Pressures within the lymphatic system are minimal, and the valves are essential to maintaining normal lymph flow towards the thoracic cavity. Contractions of skeletal muscles surrounding the lymphatic vessels aid in the flow of lymph. Lymphatic vessels are often associated with blood vessels. Differences in size, general appearance, and branching patterns distinguish lymphatic vessels from arteries and veins. We can see characteristic color differences in living tissues. Most arteries are bright red, veins are dark red, although they're usually illustrated as blue to distinguish them from arteries in the literature, and lymphatic vessels are a pale green color. In general, a tissue contains more lymphatic vessels than veins, but the lymphatic vessels are much smaller. Two sets of lymphatic vessels collect lymph from the lymphatic capillaries, the superficial lymphatics and the deep lymphatics. Superficial lymphatics are located in the subcutaneous layer deep to the skin, in the areolar tissues of the mucosal membranes lining the digestive, respiratory, urinary, and reproductive tracts, and in the areolar tissues of the serous membranes lining the pleural, pericardial, and peritoneal cavities. Deep lymphatics are larger lymphatic vessels that accompany deep arteries and veins, supplying skeletal muscles and other organs of the neck, limbs, and trunk, and the walls of the visceral organs. Superficial and deep lymphatics converge to form even larger vessels called lymphatic trunks. The trunks, in turn, empty into two large collecting vessels, the thoracic duct and the right lymphatic duct. The thoracic duct collects lymph from the body inferior to the diaphragm and from the left side of the body superior to the diaphragm. The smaller, right lymphatic duct collects lymph from the right side of the body superior to the diaphragm. The thoracic duct begins inferior to the diaphragm at the level of vertebrae L2. The base of the thoracic duct is an expanded sac-like chamber called the cisternal chyli. The cisternal chyli receives lymph from the inferior part of the abdomen, the pelvis, and the lower limbs by way of the right and left lumbar trunks and the intestinal trunk. The inferior segment of the thoracic duct lies anterior to the vertebral column. From the second lumbar vertebra, it passes posterior to the diaphragm alongside the aorta. It then ascends along the left side of the vertebral column to the level of the left clavicle. It collects lymph from the left bronchomediastinal trunk, the left subclavian trunk, and the left jugular trunk, and then empties into the left subclavian vein, near the left internal jugular vein. In this way, lymph re-enters the venous circulation from the left side of the head, neck, and thorax, as well as from the entire body inferior to the diaphragm. The right lymphatic duct is formed by the merging of the right jugular, right subclavian, and the right bronchomediastinal trunks in the area near the right clavicle. This duct empties into the right subclavian vein, delivering lymph from the right side of the body superior to the diaphragm. Blockage of the lymphatic drainage from a limb produces lymphedema. In this condition, interstitial fluid accumulates and the limb gradually becomes swollen and grossly distended. If the condition persists, the connective tissues lose their elasticity and the swelling becomes permanent. Lymphedema by itself does not pose a major threat to life. The danger actually comes from the constant risk that an uncontrolled infection will develop in the infected area. Because the interstitial fluids are essentially stagnant, toxins and other pathogens can accumulate and overwhelm local defenses without fully activating the immune system. Lymphocytes account for about 20 to 40% of circulating leukocytes. However, circulating lymphocytes are only a small fraction of the total lymphocyte population. The body contains some 1,012 or so lymphocytes with a combined weight of more than a kilogram. There are three classes of lymphocytes that circulate in the blood. You have T cells, 2, B cells, and 3, NK, or the natural killer cells. Each type has a distinctive biochemical and functional characteristics. Most lymphocytes are T cells, and the primary types of T cells include the cytotoxic T cells, or the TC cells, the helper T cells, or the TH cells, suppressor T cells, or TS cells, and memory T cells. Cytotoxic T cells are involved in a direct cellular attack. These lymphocytes are the primary cells involved in the production of cell-mediated immunity, or cellular immunity. The interplay between helper T cells and suppressor T cells helps to establish and control the sensitivity of the immune response. For this reason, these cells are also known as regulatory T cells. And we'll look at T cells throughout the rest of this module. Other types of T cells also take part in the immune response. For example, inflammatory T cells stimulate regional inflammation and local defenses as in injured tissue. Suppressor or inducer T cells suppress B cell activity, but they stimulate other T cells. Under proper stimulation, B cells differentiate into plasma cells that secrete antibodies. These antibodies are soluble proteins, also known as immunoglobulins. B cells are responsible for antibody-mediated immunity, which is also known as humoral or liquid immunity because the antibodies are also found in body fluids. Antibodies bind to specific chemical targets called antigens, which stimulate the immune response. Most antigens are pathogens, or parts or products of a pathogen or other foreign substances. Antigens are usually proteins, but some lipids, polysaccharides, and nucleic acids are also antigens. The binding of an antibody to its target antigen starts a chain reaction, leading to the destruction of the target compound or organism. Natural killer cells are also known as large granular lymphocytes. NK cells provide innate or nonspecific immunity. They attack foreign cells, cells which are infected with viruses, and cancer cells that appear in normal tissues. Their continuous policing of peripheral tissues has been called immune surveillance. The various types of lymphocytes are not evenly distributed in the blood, bone marrow, spleen, thymus, and peripheral lymphoid tissues. The ratio of B cells to T cells varies among tissues and organs. For example, B cells are seldom found in the thymus, but T cells outnumber B cells in blood by a ratio of 8 to 1. The lymphocytes in these organs are visitors, not residents. All types of lymphocytes move throughout the body. They wander through tissues and then enter blood vessels or lymphatic vessels for transport. T cells move quickly. For example, a wandering T cell may spend about 30 minutes in the blood, about 5 to 6 hours in the spleen, and about 15 to 20 hours in a lymph node. B cells, which are responsible for antibody production, move much more slowly. A typical B cell spends about 30 hours in a lymph node, a small lymph filtering structure, before moving on. Lymphocytes have relatively long lifespans. About 80% survive around 4 years, and some last even 20 years or more. Throughout your life, you maintain normal lymphocyte populations by producing new lymphocytes in your red bone marrow and in lymphoid tissues. In Module 3, we discussed hemopoiesis, which is the formation of the cellular elements of the blood. In adults, red blood cell formation, or erythropoiesis, is normally confined to red bone marrow. In contrast, lymphocyte production, or lymphopoiesis, involves the red bone marrow, thymus, and peripheral lymphoid tissues. Red bone marrow plays the primary role in maintaining normal lymphocyte populations. Hemocytoblasts divide in the bone marrow of adults to generate the lymphoid stem cells that produce all types of lymphocytes. The red bone marrow produces two distinct populations of lymphoid stem cells. One group of lymphoid stem cells remains in the red bone marrow, and the other group migrates to the thymus. Lymphoid stem cells in the red bone marrow divide to produce immature B cells and NK cells. B cell development involves intimate contact with large stromal cells in the red bone marrow. The cytoplasmic extensions of stromal cells contact or even wrap around the developing B cells. Stromal cells produce an immune system hormone, or a cytokine, called interleukin 7. It promotes the differentiation of B cells, and we'll consider cytokines and their varied effects in a later section. As they mature, B cells and NK cells enter the bloodstream and migrate to peripheral tissues. Most of the B cells move into lymph nodes, the spleen, or other lymphoid tissues. The NK cells patrol the body, moving through peripheral tissues in search of abnormal cells. The second group of lymphoid stem cells migrates to the thymus. These stem cells and their descendants develop and mature in an environment that is isolated from the general circulation by the blood-thymus barrier. Under the influence of thymic hormones, the lymphoid stem cells divide repeatedly, producing various kinds of T cells. These T cells undergo a selection process to ensure that they will not react to the body's own healthy cells and cellular products. During this process, up to 98% of developing T cells are deselected and undergo apoptosis. When the development of the surviving T cells nears completion, they re-enter the bloodstream and travel to peripheral tissues, including the lymphoid tissues and organs, such as the spleen. The T cells and B cells that migrate from their sites of origin retain the ability to divide, producing daughter cells of the same type. For example, a dividing B cell produces other B cells, not T cells or NK cells. As we'll see, the ability of specific types of lymphocytes to increase in number is crucial to the success of the immune response. Lymphoid tissues are connective tissues that are dominated by lymphocytes. In a lymphoid nodule or a lymphatic nodule, the lymphocytes are densely packed in an area of areolar tissue. In many areas, lymphoid nodules form large clusters. Lymphoid nodules occur in the connective tissues deep to the epithelial lining of the respiratory tract, where they are known as tonsils, and along the digestive, respiratory, urinary, and reproductive tracts. They are also found within more complex lymphoid tissues, such as the lymph nodes or the spleen. A single nodule averages about a millimeter in diameter. Its boundaries are not distinct because no fibrous capsule surrounds it. Each nodule often has a central zone called a germinal center, which contains dividing lymphocytes. The tonsils are large lymphoid nodules in the walls of the pharynx. Most people have 5 tonsils. A single pharyngeal tonsil, often called the adenoid, lies in the posterior superior wall of the nasopharynx. Left and right palatine tonsils are located in the posterior inferior margin of the oral cavity, along the boundary with the pharynx. A pair of lingual tonsils lies deep to the mucous epithelium covering the base of the tongue. Because of their location, the lingual tonsils are usually not visible unless they become infected and swollen. Tonsillitis is the inflammation of the tonsils, especially in the palatines, although the other tonsils may also be infected. Tonsils reach their largest size by puberty and then begin to atrophy. The collection of lymphoid tissues that protects the epithelium of the digestive, respiratory, urinary, and reproductive systems is called the mucus-associated lymphoid tissue, or the MALT. Clusters of lymphoid nodules deep to the epithelial lining of the intestines are known as aggregated lymphoid nodules, or Peyer's patches. Other examples of MALT include the appendix and the tonsils. The appendix, or the vermiform appendix, is a blind pouch that originates near the junction between the small and large intestines. Its walls contain a mass of fused lymphoid nodules. Fibrous connective tissue capsules separate lymphoid organs, the lymph nodes, the thymus, and the spleen, from the surrounding tissues. Lymph nodes are small lymphoid organs ranging in diameter from 1 millimeter to 25 millimeters. The greatest number of lymph nodes is associated in the neck, armpits, and groin, where they defend us against bacteria and other invaders. A dense connective tissue capsule covers each lymph node. Bundles of collagen fibers extend from the capsule into the interior of the node. These fibrous partitions are called trabeculae. The typical lymph node is shaped like a kidney bean. Blood vessels and nerves reach the lymph node at a shallow indentation called the ileum. 2 sets of lymphatic vessels, afferent lymphatics and efferent lymphatics, are connected to each lymph node. Afferent, from the root word afferens, which means to bring to, lymphatics bring lymph and to the lymph node from the peripheral tissues. The afferent lymphatics penetrate the capsule of the lymph node on the side opposite of the ileum. Efferent, from the root word efferens, which means to bring out, lymphatics leave the lymph node at the ileum. These vessels carry lymph away from the lymph node and towards the venous circulation. Lymph from the afferent lymphatics flows through the lymph node within a network of sinuses, which are open passageways with incomplete walls. Lymph first enters a subcapsular space, formerly called the subscapular sinus. It contains a meshwork of branching reticular fibers, macrophages, and dendritic cells. Dendritic cells are involved in starting an immune response, and we'll consider their role a little bit later as well. Lymph passes through the subscapular space and then flows to the outer cortex of the node. The outer cortex contains B cells within germinal centers similar to those of lymphoid nodules. Lymph then continues to the lymph sinuses in the deep cortex, or the paracortical area, which is dominated by T cells. Here, lymphocytes leave the bloodstream and enter the lymph node by crossing the walls of the blood vessels. After flowing through the sinuses of the deep cortex, lymph continues into the core, or medulla, of the lymph node. The medulla contains B cells and plasma cells organized into elongate masses known as medullary cords. Lymph passes through a network of sinuses in the medulla and then enters the efferent lymphatics at the hilum. A lymph node functions like a kitchen water filter. It purifies lymph before it reaches the veins. As lymph flows to a lymph node, at least 99% of the antigens in the lymph are removed. Fixed macrophages in the walls of the lymphatic sinuses engulf debris or pathogens in lymph as it flows past. Antigens removed in this way are then processed by the macrophages and presented to nearby lymphocytes. Other antigens bind to receptors on the surfaces of the dendritic cells, where they can stimulate lymphocyte activity. This process, or antigen presentation, is generally the first step in the activation of the immune response. In addition to filtering, lymph nodes function as an early warning system. Any infection or other abnormality in a peripheral tissue puts antigens into the interstitial fluid and thus into the lymph leaving the area. These antigens then stimulate macrophages and lymphocytes in nearby lymph nodes. To protect a house against intruders, you might guard all the entrances and exits or place traps by the windows and doors. The distribution of lymphoid tissues in lymph nodes follows such a similar pattern. The largest lymph nodes are located where peripheral lymphatics connect with the trunk, such as in the groin, the axillae, and the base of the neck. These nodes are often called lymph glands. Because lymph is monitored in these nodes, potential problems can be detected and dealt with before they affect the vital organs of the trunk. These mesenteries of the gut also have aggregations of lymph nodes located near the trachea and the passageways leading to the lungs and then associated with the thoracic duct. These lymph nodes protect against pathogens and other antigens within the digestive and respiratory systems. A minor injury commonly produces a slight enlargement of the nodes along the lymphatic vessels, draining the region. This sign is often called swollen glands. It typically indicates inflammation in peripheral structures. The enlargement generally results from an increase in the number of lymphocytes and phagocytes in the node in response to a minor localized infection. Chronic or excessive enlargement of lymph nodes is lymphadenopathy. This condition may occur in response to bacterial or viral infections, endocrine disorders, or cancers. The thymus is a pink, grainy organ located in the mediastinum, generally just posterior to the sternum. In newborn infants and young children, the thymus is relatively large. It commonly extends from the base of the neck to the superior border of the heart. The thymus reaches its greatest size relative to body size in the first year or two after birth. The organ continues to increase in mass throughout childhood, but the body as a whole grows even faster, so the size of the thymus related to that of other organisms in the mediastinum gradually decreases. The thymus reaches its maximum absolute size at a weight of about 40 grams just before puberty. After puberty, it gradually diminishes in size and becomes increasingly fibrous, a process called involution. By the time a person reaches age 50, the thymus may weigh less than 12 grams. The gradual decrease in the size of functional tissues and secretory abilities of the thymus may make elderly people more susceptible to disease. The capsule that covers the thymus divides into two thymic lobes. Fibrous partitions called septa originate at the capsule and divide the lobes into lobules, averaging about 2 millimeters in diameter. Each lobule consists of a densely packed outer thymic cortex and a paler central medulla. The cortex contains actively dividing lymphocytes, or T cells. As the T cells mature, they migrate into the medulla. After about 3 weeks, these T cells leave the thymus by entering one of the medullary blood vessels. Lymphocytes in the cortex are arranged in clusters that are completely surrounded by the thymic epithelial cells, also called TECs. Thymic epithelial cells regulate T cell development and function. Epithelial cells also encircle the blood vessels of the cortex. These cells maintain the blood-thymus barrier. Maturing T cells leave the cortex and enter the medulla of the thymus. The medulla has no blood-thymus barrier. The thymic epithelial cells in the medulla cluster together in concentric layers, forming distinctive structures known as thymic corpuscles. Despite their imposing appearance, the function of the thymic corpuscles remains relatively unknown. T cells in the medulla center can leave the bloodstream across the walls of blood vessels in this region or within one of the efferent lymphatics that collect lymph from the thymus. The thymus produces several hormones that are important to the development and maintenance of normal immunological defenses. Thymosin is the name originally given to an extract from the thymus that promotes development and the maturation of lymphocytes. This extract actually contains several complementary hormones. They include thymosin A, thymosin B, thymosin V, thymopoietin, thymulin, and others. The plural term, thymosins, is now sometimes used to refer to all of the thymic hormones. The adult spleen contains the largest collection of lymphoid tissue in the body. In essence, the spleen performs the same functions for blood that lymph nodes perform for lymph. Function of the spleen can be summarized as 1. Removing abnormal blood cells and other blood components by phagocytosis, 2. Storing iron recycled from blood cells, and 3. Initiating immune response by B cells and T cells in response to antigens and circulating blood. The spleen is about 12 centimeters long and weighs on average nearly 160 grams. In gross dissection, the spleen is deep red due to the blood that it contains. The spleen lies along the curving lateral border of the stomach, extending between the 9th and 11th ribs on the left side. It's attached to the lateral border of the stomach by the gastrosplenic ligament, a broad band of mesentery. The spleen has a soft texture, so its shape primarily reflects the shapes of the structures around it. The spleen is in contact with the muscular diaphragm, the stomach, and the left kidney. The diaphragmatic surface is smooth and convex, conforming to the shape of the diaphragm in the body wall. The visceral surface contains indentions that conform to the shape of the stomach, or the gastric area, and the kidney, or the renal area. Splenic blood vessels, or the splenic artery and the splenic vein, and lymphatic vessels communicate with the spleen on the visceral surface at the hilum, which is a groove marking the border between the gastric and the renal areas. The spleen is surrounded by a capsule containing collagen and elastic fibers. The cellular components within make up the pulp of the spleen. Red pulp contains large quantities of red blood cells, and white pulp resembles lymphoid nodules. The splenic artery enters at the hilum and branches to produce a number of arteries that radiate outward towards the capsule. These trabecular arteries in turn branch extensively, and their finer branches are surrounded by areas of white pulp. Capillaries then discharge the blood into the red pulp. The cell population of the red pulp includes all of the normal components of circulating blood, plus fixed and free macrophages. The structural framework of the red pulp consists of a network of reticular fibers. The blood passes through this meshwork and enters large sinusoids, also lined by fixed macrophages. The sinusoids empty into small veins, which ultimately collect into the trabecular veins and continue towards the hilum. This circulatory arrangement gives the phagocytes in the spleen an opportunity to identify and engulf damaged or infected cells in circulating blood. Lymphocytes are scattered throughout the red pulp, and the area surrounding the white pulp has a high concentration of macrophages and dendritic cells. For this reason, any microorganism or other antigen in the blood quickly comes to the attention of lymphocytes. The spleen tears so easily that a seemingly minor hit to the left side of the abdomen can rupture the capsule. The result is serious internal bleeding and the eventual circulatory shock. Such an injury is a known risk of contact sports, such as football, hockey, and rugby, and of more individual athletic activities, such as skiing and sledding. Because the spleen is so fragile, it's very difficult to repair surgically. Sutures typically tear out before they've been tensed enough to stop the bleeding. A severely ruptured spleen is removed, a process called a splenectomy. A person can survive without a spleen, but lives with an increased risk of bacterial infection, particularly involving pneumococcal bacteria. Ruptured spleen is one of the biggest risks for the disease mono as well. And as we've noted, the human body has multiple defense mechanisms. Together they provide resistance to the natural or acquired ability to maintain immunity. And we can sort the body defenses into two general categories. One, the innate nonspecific immunity does not distinguish one type of threat from another. Their response is the same regardless of the type of invader. These innate defenses are present at birth, and they include the physical barriers, phagocytotic cells, immune surveillance, interferons, complement, inflammation, and fever. They provide a defensive capability known as nonspecific resistance. Two is the adaptive or specific immunity, which protects against particular threats. For example, an adaptive defense may protect against one type of bacterium, but not another type of bacteria and virus. Many specific defenses develop after birth as a result of accidental or deliberate exposure to environmental hazards. Adaptive defenses depend on the activities of specific lymphocytes. B cells and T cells are part of our adaptive defenses, which provide protection known as specific resistance. Innate and adaptive defenses work together. Both must function normally to provide adequate resistance to infection and disease.